Okay, this is our Meselson-Stahl verbal video. And then, so basically, a little background to our experiment, to their experiment. Um, they put the E. coli bacteria into the N15 medium, and it's really heavy. So then, as they grew the cells in the N15 medium and waited for all the DNA to have the N15, um, all the DNA were heavy, and then they transferred that into the N14 medium, which is a lighter nitrogen. And then their entire experiment is based on the DNA replication in the N14 medium. And as the DNA replicates in the N14 medium, they had three different hypotheses. One's conservative, the second one's semi-conservative, and the last one is dispersive replication. And then they use evidence through centrifugation to separate the heavier N15 gene from the lighter N14 gene from density. And then to draw their conclusions, they relied on the presence, the amount of bands, and their relative locations in the tubes. And the heavier N15 would be on the bottom. The hybrid N15 plus N14 would be around the middle. And then the lightest N14 would be on the top.
If the conservative hypothesis were true, it would mean that it would be separated into two strands because there would be no interaction between the new strand and the parental strand. In the hypothesis, the replication strand would replicate the new strand of DNA off of the lighter isotope, but still leave the parental strand, the heavier isotope, intact. Therefore, it will separate both the heavier and lighter isotopes, meaning that one molecule will be all light, and the other one all heavy.
Okay, and if the dispersive results were true, the first generation N15 should separate and kind of mix with the N14 DNA, and it will form the pattern shown in this diagram in generation 2. And if you continue this logic, the hybrid mix of N15 and N14 from this generation will rearrange again with the pure N14 DNA from the medium to produce more mixes of the N15 and N14 in the third generation. And as they are all hybrid DNAs, the bands should appear in around the same area. However, there would be multiple bands in that area because there could be many possibilities of N15 to N14 ratio that create different mixtures of masses.
And here's why conservative and dispersive would be incorrect. Conservative would be an incorrect replication because the first generation of the experiment showed that when the light and heavy DNA were spun in the centrifuge and separated by weight, it showed that half of the DNA contained heavy nitrogen, N15, and the other half did not. This is not in line with the conservative replication because its model predicts that one molecule would be completely N15 and the other molecule would be completely N14. Dispersive would also be incorrect because in the data shown in Meselson and Stahl's experiment, there were two distinguishable bands, a thin one for hybrid and a thick one for pure N14. In the dispersive experiment, only the hybrid DNA was present.
Semi-conservative replication is actually true because the lighter isotope, N14, would consistently replicate, but the original parental strand still remained, which mixed with the new DNA, resulting in a medium strand. Every cell gets one old DNA strand and one new one, meaning that the DNA strands separated. This will result in the mixed DNA. However, over time, a new band of the lighter isotope will form because of the lighter isotope outpacing the parental DNA.